Good, so welcome everyone. My name is Vicente Barot, and in the LIBREhub project, we organize workshops and seminars to help scientists use open hardware tools for bioimaging. So in our seminar today, we welcome Rebecca McClelland. She's a PhD student at the University of Cambridge who is working with Klemens Kaminsky, and she will tell us about her project, about the simple microscope. She has been developing a structured illumination microscope to use in unconventional environments. So thank you very much, Rebecca, for joining, and the floor is yours.
Thank you. Thank you so much for having me in your seminar series, and thank you everyone for listening. So as Vicente said, I'm a PhD student in the laser analytics group at the University of Cambridge. And I've been working on optical microscopy development during my PhD. I'd just like to tell you a bit about the simple microscope that I have been working on. So in my presentation, I'll just quickly recap what SIM is, the different types of SIM that there are, the motivation for this project, go over the design that we worked on, and then how we implemented that design into different instruments, and then finally to discuss about how this could help improve accessibility to SIM in line with the seminar's ethos. So first of all, what is structured illumination microscopy? So SIM is a fluorescence imaging technique that can achieve optical sectioning and/or super resolution imaging. It can achieve theoretically up to 2 times the resolution of standard widefield imaging. And the way that it does this is by illuminating our samples with multiple stripe patterns that you can see here. And these multiple images are captured using widefield detection and then can be post-processed into our higher contrast or higher resolution image. So a lot of the development that goes on with SIM relates to the types of patterns that are generated and also the hardware to generate those patterns. So in my presentation, I'm just going to consider 3 or 9 sinusoidal patterns, but there are a range of different illumination schemes that are used. So on the right here is an example of what a typical implementation based on an SLM or a spatial light modulator might look like. In addition to some sort of static optics for combining the beams and polarization control, most of the control is done with the SLM that we can see at the top here. And essentially what this does is it splits your coherent laser beam into 2 or 3 beams, changes the relative phase of those beams, rotates them around the optical axis in order to achieve the rotation that you can see in the different rows here, and then finally overlaps them at the sample. So those first 2 steps are all done by the SLM itself, and then we just have a series of lenses in a 4F relay to relay those beams from the SLM to the objective lens, where you can see that they cross over at the sample. And so the stripe patterns are generated by the constructive and destructive interference of the 2 beams. And so what are the types of SIM? So as I said, there are quite a few types, but in my presentation today, I'm just going to focus on these 2 here. So the first is optical sectioning, which requires 3 more frames. So as you can see in the middle here, our sample is illuminated with a stripe pattern. And for the purposes of sectioning, what we're interested in is the fact that when the sample is in focus, so in the focal plane of the objective, we can see that there are stripes that are visible, indicated by the orange arrow. But in the thicker nucleus region, indicated by the blue arrow, we can't see any stripes because it's out of focus. And essentially, the SIM reconstruction process identifies these 2 areas and only maintains signal that is in focus, leading to this reconstructed image that you can see on the right-hand side. And then a second type of SIM, super resolution SIM. This is what we're usually referring to if you just say SIM by itself, can increase the resolution. So we have 9 illumination patterns, as you can see in this GIF in the middle, 3 stripe patterns shifted from side to side or shifted in phase, and then also rotated and shifted in phase again. And when we reconstruct these images, we get something on the right-hand side. And as you can see, compared to the wide field image, the resolution is higher. And theoretically, this can be up to a 2 times increase, but 1.7 to 1.9 is typical. So this can achieve a resolution down to 100 nanometers, which is obviously a lot better than the basic wide field case. So that's of interest where you have sort of small details that you need to visualize in your sample. And optical sectioning can be advantageous where you want imaging comparable to confocal, but at faster acquisition speeds. So that makes it suitable for imaging live samples, unlike confocal, which sometimes can be a bit slow for live organelle dynamics. So what was the motivation of this project? So SIM has many advantages for biological research, chiefly being that it is ideally situated as a trade-off between resolution and imaging speed. We can achieve resolution on the 100 nanometer scale, but at the same time, we can image fast enough and with gentle enough laser power that we can observe live cells over a long period of time. And we also have the ability to image multiple fluorophores, so we can label different organelles within the cells with different labels to identify them. And finally, although the reconstruction process to turn those raw images into the final output image is quite complex, there are lots of great open access software tools for image processing, which you can integrate with ImageJ for really simple reconstruction through an accessible GUI, making it easy to use. However, the flip side of this, so in terms of imaging performance, SIM has achieved a lot. However, we think that sometimes the disadvantages of this technique are its accessibility. If you wanted to use this technique in your own lab, you're looking at a very hefty price tag to just buy a commercial system off the shelf. And alternatively, if you wanted to build it yourself from the ground up, this is slightly cheaper, but requires a lot of optics knowledge and experience, and also a big time investment to learn how to build this and to build it. And then once you have your instrument, you need a lot of space in order to house it. You need to control the environment very carefully, particularly with regard to vibrations, often requiring an optical bench with vibration isolation, which are also quite expensive. And so it's just generally very sensitive to its environment. When using a SIM, if you want to perform long-term live cell imaging, typically you need some kind of stage top incubator to maintain physiological condition for your samples, whilst the microscope itself needs to be housed in ambient conditions, as I've discussed. So this can present further engineering challenges. And finally, you can avoid the problem of buying or building your own SIM by using the instruments provided in an imaging facility or a core facility. But then this comes with the trade-off of how can you transport your delicate samples to the facility, and how can you set up your experiment outside of your own lab where all of your equipment and resources is. So there are lots of factors to consider. Even if you might like to use SIM for your experiment, there are lots of factors to consider of how you can use it. So in our project, what we wanted to do was to maintain the imaging performance of current SIM instruments, but also consider the factors like the complexity, robustness, and footprint, which currently limit its accessibility. So the way that we approached this was to develop a new optical design for the illumination path. So this is our optical design for SIM, and the main way that we achieved a much lighter and easier to assemble instrument was to utilize inline fiber components. So in this method, our manipulation steps of the beam are achieved by separate fiber components. So first of all, our multiple laser lines are coupled into an input of a fibre-based splitter. So the light is traveling along fiber patch cables, which then connects to this device, which splits the light into 2 fibre outputs, each of which have 50% of the power. One of these outputs is connected to an inline phase shifter, and basically this can control the phase with incredible precision of the light simply by applying a voltage to the device. So we now have a phase shift between the 2 arms of the interferometer, which are then output here at the fibre array, which allows them to be output very close together, perfectly parallel. And this fibre array is housed inside a rotation mount, which allows us to achieve rotation of the pattern. So the phase shifting is achieved by the phase shifter and the rotation by the rotation mount. And then the fibre array, in combination with these free space optics that you can see at the bottom, relay the light to be incident on the back focal plane of the objective in order to create our sim patterns. So the first advantage of this is the cost. So assuming you already have a wide field microscope with your lasers, an objective and detection path, the only additional cost that you would need to upgrade that wide field microscope into a sim microscope come at a total cost of $3,500 US, which is obviously definitely at the cheaper end for sim systems. But more importantly than cost, what we would like to say is that it's incredibly easy to assemble. So if you look at this image on the left here, this is a photo of the fibre components in the diagram. I don't know if you can see my mouse. So at the bottom here, this red component is the splitter. So light is input there and then exits to the 2 outputs, which are in red and white, with the first output being connected to the fibre phase shifter, which you can see in the top left, and the second output just being connected to a patch cable, both of which connect to the array. And as you can see from the animation in the top right-hand side, it's incredibly easy to put these components together. No optical alignment required. You just plug and play and they're ready to go. Similar connections can be used to connect for the fibre phase shifter, which is not shown in the left-hand picture. And then in the bottom right-hand picture, which I hope the animation is appearing correctly, shows, so from the right-hand side, these yellow patch cables, we can choose which of these cables to connect with to the rest of the circuit. And then this will determine which of the outputs the light is emitted from. And so we can just choose a single central output if we want wide field imaging, or we can choose different separations of 2 beams if we want optical sectioning or super resolution imaging. In this diagram, there are just 5 outputs for clarity, but in reality, we have 9 different outputs. And because the spacing between these is only 127 microns, we have incredibly fine and precise control over the position that these beams are exiting the fibre array, which in turn gives us control of the spatial frequency of our illumination patterns. So this makes the system compatible to work with any objective lens and the 2 different imaging modes. We just choose which patch cables to connect to get our desired output. So that's the concepts behind the design. So the first implementation that we did for this was an add-on module. So this is assuming that you already have a wide field microscope set up in your lab. This is how you can upgrade it to a sectioning sim instrument. So you will just need to couple your light sources into the fibre splitter. And then this is the add-on module. So from the right-hand side to the left-hand side, we have the fibre array mounted in this grey 3D printed jacket, which is in a Thorlabs cage-mounted optomechanic. Then we have a collimating lens, a pinhole, and a polarizer. And then this is the tube lens on the far left. And at the bottom here in green, you can see how it can connect to the rest of the fibre circuits. Again, just using these mating sleeves, which take sort of 10 seconds just to plug in. So first of all, the module is incredibly compact. The total path length is 300 millimeters, and it's 60 by 60 millimeters. not including the post, that you'll just need to mount it to the correct height for your system. So even with very limited space, it's quite easy to fit this in to your workflow. And the fibre array only needs to be aligned for tip tilt and in X and Y. But once that bit is done, the use of the cage system means that everything else fits in very easily and only needs to be aligned in the Z direction, where Z is the optical axis going from right to left in this picture. So we say that even if you're not an expert at optics, it is possible for you to build this due to the fibre-based design, making it a lot easier to implement. So we then demonstrated the system by imaging some fixed cells. So what you can see for comparison here is a wide field image on the left and the optical sectioning image on the right. So these two images are captured using the same objective lens and the same camera. But what we can see from using the sectioning sim is that we have a much clearer picture of the Z-axis, that the 5 images have been captured in Z. And not only are the microtubules easier to resolve, but also if we were to do a 3D projection, we could see the sort of the hill of the nucleus in the center, and it's the image is much clearer, mapping the different heights in the sample. We also imaged live cells. So for the sectioning imaging, the system is incredibly fast. The phase shifter has a 20 kilohertz response. So essentially that means 0.05 milliseconds delay between each of the 3 raw frames you need to capture. So essentially your system is only limited by how fast you can generate the voltages, which again is much faster than a typical exposure time. So essentially making the system camera limited. And as you can see, again, from a wide field on the left for a sectioning sim comparison on the right, is that we can far more clearly visualize the mitochondria in a COS7 cell for better visualization of the dynamics. And we're also able to do multicolor imaging. So an advantage of using the fibre array output is that all of the wavelengths are emitted exactly coaxially. So they will all be incident on the same points in the back focal plane of the objective, meaning that we can generate any wavelength simultaneously with the correct spatial frequency of the illumination patterns. And here we've demonstrated that by imaging the mitochondria and the endoplasmic reticulum in our system. And these short videos should show that we can, well, first of all, is that we can say with confidence about the interactions between the organelles because we know that the images were captured exactly at the same time. There's no delay between the color channels. And then, as you can see with the sectioning sim, you can visualize far more clearly what's going on. So these images were captured with 491 and 561 nanometer illumination. And so our second implementation of this design was for super resolution imaging. So rather than just providing a DIY add-on, we wanted also to create a complete self-contained instrument that didn't require any extra parts. And that's what you can see in this photo and the render here. So this is the same optical design. So light comes in from the fibers as we've seen before. The only difference being that this section here, which is the same as the add-on module we saw previously, has a rotation mount which rotates the cage system. So this generates the rotation of our patterns needed for super resolution imaging. And as you can see from the fact that I'm no weightlifter, but I can just hold it very easily, it's very lightweight and it's also very compact. So a single person could very easily just pick this up, put this in the boot of their car and take it wherever it needs to go. We have implemented the system with 2 different sample mounting systems depending on requirements. So on the left, we have a completely automated X, Y, and Z control. So if you needed to be able to control the microscope remotely, you could do so in that way. And on the right-hand side, the render shows sample mounting using a nose piece, which very effectively isolates the objective and the sample from environmental noise or any noise coming through the breadboard. So this gives the instrument excellent stability if you want to image in an environment where environmental control is limited and you're expecting a lot of noise surrounding the system. And finally, just to say that it's very compact, 300 by 300 by 450 millimeters. So very easy to fit this on in your lab space. Yeah, very easy to fit in your lab space. We then demonstrated super resolution imaging of fixed cells with this system. So again, we have a wide field comparison using the same objective and the same camera. And what we see from these two comparisons in the middle here is that we can far more clearly visualize the microtubules with the resolution increase that we get from doing super resolution sim. This was found to be 167 nanometers based on decorrelation analysis of this image on the left. And on the right-hand side shows the results of just analyzing beads with the system, looking at the full width half maxima of sub-diffraction beads. And this found that the resolution increase was 1.91 compared to wide field imaging for the 561 nanometer wavelength, and it was 1.92 with the 491 nanometer wavelength. And again, like with the sectioning, we're able to generate those patterns simultaneously, although currently we can only image with one color due to not having an optical split system that we saw in this previous version. So how do we propose to improve accessibility to SIM? So in terms of technological development, that's what we've already done and I've talked about in my slide. We've got multicolor live sectioning imaging and also super resolution imaging of fixed cells with a 1.91 times resolution increase. We can image across a incredibly large field of view, over 100 microns, and this is only limited by the physical diameter of our lenses and camera chip in the system. And we demonstrated a cheap, compact, easy to assemble sim design. So the next step will be to further utilize fiber technology and use a two-dimensional output array instead of a one-dimensional output array in combination with fiber switches. So this will bring the speed of the sectioning, bring the speed of the super resolution imaging up to be also camera limited in the same way that the sectioning is. But then, perhaps more interestingly to you, we'd like to use this design to improve access to SIM. So as previously discussed, we think there are lots of limitations for accessibility. And although there has been some excellent work done to address this in terms of open source designs, we think that sometimes these can overestimate the time and skill set of the users, or rather the area of the skill set of the users, as very well demonstrated by this XKCD comic. I think for microscopy developers, we tend to take for granted that things like optical alignment, laser machining, soldering circuit boards, etc., are skills that everyone has, but actually these aren't some things that everyone knows how to do and are not necessarily the skills that people are interested in developing, you know, when they've got, for example, a huge range of wet lab skills that they've already learned and want to use. So we think sometimes open source designs can fall short in that regard, in that they just take, even though the price list is very low, they're not always feasible for everybody to replicate. And again, maintenance of physiological conditions over long periods and transportation of samples, as I previously discussed. So we'd like to address those problems in several ways. So first of all, in our previous publication, we've already made our optical sectioning reconstruction code completely open source and available, because most of the open source code available to the public is for super resolution reconstruction. So we've already made that completely open source for the machine learning networks. Shortly, hopefully by the time this is on YouTube, should be out, we'll have a preprint which will present complete details of our design for the add-on module. What we uniquely propose with this is that, with one exception, absolutely everything can be purchased off the shelf. And as you've seen with the fibre connections, is very easy to assemble. It's mostly just plug and play. And where alignment is required, this is massively simplified by using a cage system with only a few pieces that need to be aligned. And all of that can be done with just two pieces of equipment, a t-shirt target and a shearing interferometer. And we'll make all those details available in a way that we think realistically a non-expert optics user could replicate. We also say that if you don't want to 3D print your own jacket for the fibre array, we also, in addition to a 3D printed version, made one in our mechanical workshops from brass. So if you would like to contact us, we can probably just send you the version, so you don't even need to worry about manufacturing that yourself. So secondly, so obviously not everyone has the time to build a microscope from scratch, even if the design is really easy. So we were very inspired by the Flamingo light sheet model proposed by the Huisken lab. And so what they propose is actually we'll just loan you the instrument itself. And we think that the Simple's compact, lightweight and robust design lends itself very well to this application. So we propose that we could collaborate with other groups to offer short-term loans of a couple of weeks. We can bring the microscope to you, set it up, and show you how to use it. And that way you have access to this technique from the comfort of your own lab, but without having to buy or build one yourself. So hopefully thereby lowering the cost and expertise barrier. Finally, we also hope to enable imaging in unconventional environments, as said in the title. And what we mean by that is currently the need for stage-top incubation can be a limiting factor in long-term live cell imaging experiments. Now, previous work has addressed this by building an entire microscope that can fit inside an incubator. But most of these implementations are only widefield implementations. They're not capable of super resolution imaging. So what we propose is that we can do super resolution imaging in the incubator by just fitting the entire frame of the instrument inside, be it a standard warm incubator or the freezer incubators used by the British Antarctic Survey for looking at Antarctic species. So we think that this could enable better long-term control of the cell environment, because both your cell sample and the entire microscope is being kept in these conditions. And this is enabled by the fibre design, which means that you don't need to put your delicate and expensive spatial light modulator in a very high humidity environment where that could end quite badly for you. And finally, we also, as I've just said, the fibres house your delicate pattern generating optics and your expensive lasers away from the mainframe of the microscope again, so they can be kept safely in ambient conditions. But your microscope, for example, the objective, which is in contact with the sample, can be kept in the same conditions that you need for your sample. So enabling long-term imaging experiments in that regard. And this would also help with decontamination of the optics. Again, limiting the number of pieces of equipment that need to be decontaminated, for example, if you're imaging in a BSL 3 or 4 environment. So thank you very much for listening. I'd like to say a quick thank you to my supervisor, Clemens Kaminski, Edward Ward, who's helped with the design, building and imaging with the system, Francesca for sample preparation and being our resident biology expert, and also Stephen and Junqing, who worked on the construction and characterization of earlier versions of the microscope. And thank you to AR UK for a pump priming award that supported this work. And thank you to you for listening.
Thank you very much, Rebecca. That was very interesting.